Hello everyone, welcome back to my YouTube channel, Life Sciences by Mutations. This is an online teaching platform where I give lessons, full-on lessons on life sciences material. So far I've been doing content based off of grade 12 work. Soon and very soon I will be doing content on grade 11. If you are new to my channel or if you are a returning friend, please do react to my video by pushing the like button, comment any content that you would like me to record next, subscribe to the channel so that my lessons do not get lost in your YouTube algorithm, and please do share the link to this video to other people that might benefit from the lessons that I am providing. My name is Mutations. I am a life sciences teacher at Milner High School. This will be lesson 7, dealing with mutations as well as DNA profile. This is the last of 7 long videos on the DNA code of life. Before we get on with the video, let us check first what is it that we've been doing in the previous lessons and what is it that we are going to do going forward. This is an outline of how I decided to structure my lessons on the topic of DNA, the code of life. On lesson 1, we looked at the revision of the structure of the cell. Lesson 2, we looked at the DNA structure. Lesson 3, we looked at DNA replication. Lesson 4, we looked at the history of the discovery of DNA. On lesson 5, we looked at the second nucleic acid, which is RNA. We looked at its structure. Lesson 6 was a 2-part lesson on protein synthesis. Lesson 1 was based on the process of protein synthesis, which is divided into transcription, movement of mRNA, as well as translation. On the second part of the very lesson, we dealt with amino acid coding. And on this lesson, this is lesson 7, which is based on DNA profiling. We are going to be looking at the definition and the uses of DNA profiling, as well as the factors that affect the reliability of DNA profiling. We will be doing some interpretive work, interpreting different DNA profiles in order to determine paternity from a set of potential fathers. And then we will also be looking at DNA profiles dealing with forensic evidence in a criminal case. So, if you're interested in if you're new to my channel and you are interested in going back and looking at the full-length videos on each of these subtopics, you are welcome to do that. Okay, let's get right into the lesson. Since this is the last lesson on the video series of the DNA code of life, I just deemed it important to let you know that the topic of the DNA code of life will be in your paper 2, remember, at the end of the year. And in September, when you are writing your preparatory exams, you are going to be writing 2 papers, each constituting 150 marks. The topic of the DNA code of life is going to be in your paper 2, and then it's going to be 18% of the paper, and then it's going to constitute 27 marks. So all the lessons that we've been dealing with, all the exam type questions that I've been giving you, you might not get the same type of questions. No. You might not get the same exact question. However, if you master the skill of answering each exam type question in each activity that we've done in the previous lessons, then you are good and well to get these 27 marks. Okay, should you have any question or encountering a question as you are going through previous question papers or activities that you're doing at your particular school, please do post that in the comments, and then I will help you out with that question. Okay. In the first part of this lesson, we will be looking at mutations. A mutation. We need to define what the term mutation means. So when you speak about a mutation, we are talking about a random error that changes the structure and function of the DNA code. Okay? It is an error, meaning it is a mistake that happens, and that mistake is quite often random. Okay? You are not required to know the different causes of these random mistakes that change the structure and that change the function of DNA. The causes are not prescribed for you in your exam guidelines. So the definition of a mutation, like I said, to go back, is that it is a random error that changes the structure and function of the DNA code. Now, we have two types of these errors, okay? But before we go to the two types of mutations that are prescribed for your grade 12 work, these errors may take place during DNA replication when DNA does not form an identical copy. Remember on lesson 3, we dealt with DNA replication. We said that DNA replication is a process where DNA makes identical copies of itself. Now, if DNA does make a copy, but the copy is not identical, then that may result in a mutation. Or during protein synthesis, the wrong DNA code is transcribed, which affects the mRNA codon, which will affect the tRNA anticodon, which will also affect the sequence of amino acids in the polypeptide chain, and that will cause a wrong protein to be made or a different protein to be made. Okay, we've looked at what happens if the wrong DNA code is transcribed in the previous lesson. So please go and have a look at that. We looked at how that happens and how it affects the synthesized protein. Or a mutation can take place during cell division, where the mother cell produces daughter cells that contain more or less chromosomes than should be. Okay, so these are where the mutations or the errors may take place. It is not prescribed for you to know this. This is just pre-information that I'm giving you for context of this lesson. Okay, but what you are required to know are the two types of mutations. Firstly, we have what we call a gene mutation, and we have what we call a chromosomal mutation. What is the difference between the two? In terms of a gene mutation is a change in the sequence of nitrogenous bases of DNA. This also you are required to know. So if there is a change in the sequence of nitrogenous bases on DNA, we call that a gene mutation. What do we mean by that? Okay, let's say, for example, our DNA template reads A, G, okay, excuse me for that. If our DNA code should read A, C, T, okay? But instead of ACT, there is a mistake that happened, and then our DNA code, let's say during DNA replication, instead of making ACT, the process of DNA replication makes now A, T. Okay, so as you can see from this DNA template ACT, there was an error that was made. So instead of ACT, the wrong template was made, and now it reads AAT. Okay, of course, this is going to have implications in terms of the mRNA codon that is made. The mRNA codon now will be, let's check, it will be U, A, instead of U, G, A. Okay? And if the mRNA codon changes, then it means that the tRNA anticodon will also change. The amino acid that is brought to the ribosomes during translation will also change, and a wrong protein will be made. Okay, instead of the protein that initially had to be made by the cell. Okay, so this is a gene mutation. Why? Because it is a change in the sequence of nitrogenous bases on DNA. Okay, so this results in an error or a mutation that we refer to as a gene mutation. However, we have more serious mutations that we call chromosomal mutations. Chromosomal mutations are also referred as chromosomal aberrations, but your exam guidelines specifically use the term chromosomal mutation, so that is the terminology that I'm going to stick to. Okay, now in humans, human cells, let's say for example, this is a human cell, and inside the human cell we have a nucleus. And then from our previous lessons, we had mentioned that a human somatic cell must contain 46 chromosomes. Okay? It must contain 46 chromosomes. Now, if we have more of chromosomes within the cell, or we have less chromosomes within the cell, that we call as a chromosomal mutation. Okay, so a chromosomal mutation is a mutation that produces cells with either more or less the number of chromosomes than normal. Okay, like I said, a normal somatic cell must have 46 chromosomes. However, if we have more than 46 or less than 46, we refer to that as a chromosomal mutation. Okay, so chromosomal mutations affect the number of chromosomes that are in a cell, and then a gene mutation changes the sequence of nitrogenous bases on DNA. Okay, guys, let's move on to the next slide. So, what are the effects of mutations? What do they cause? Okay, we have two types of effects of mutations that are prescribed for you in grade 12. We have harmless mutations as well as harmful or lethal mutations. So we have mutations that are harmless. If that mutation occurs in the DNA, the effect on the human body will be harmless. Okay. Let's firstly start, however, with harmful or lethal mutations. Usually, they cause a genetic disorder such as cancer. Okay, on that particular tissue or that particular cell or that particular area of the body, if we have a mutation and that mutation is harmful, it may cause genetic disorders that may lead to conditions such as cancer. Okay, of course, cancer is harmful, and remember, the word lethal means that it might kill you. Okay, so it might lead to mortality. More about harmful mutations. They may compromise the life of the organism and may lead to death, hence the word lethal. Okay, so harmful mutations or lethal mutations, they compromise the life of that particular organism. In terms of human beings, they are going to cause certain compromises to your health, certain compromises to the way that you live your life, and if left untreated, they might lead to death. Hence, they are lethal mutations. However, harmless mutations do not cause any disorder to the organism. Okay, they are harmless. So there is no harmful effect to them. So they have no negative impact. So no negative impact is experienced by the organism. In actual fact, the organism might not even notice that the mutation is there. Okay, so they do not cause any disorder, hence they are harmless. They might not have any negative expression on the body of the organism as well. So with these types of mutations, we have two more effects. One is that there might be a neutral effect. We call those neutral mutations, or they might be beneficial. Okay, the effect might be beneficial, meaning providing something that is going to add value to your life. We call those beneficial mutations. With neutral mutations, these are also called silent mutations. They are usually not expressed. By expressed, we mean they are not shown on the organism's body. So they are only seen or experienced on the genes or on the DNA of the organism. However, they are usually not expressed or seen on the body of the organism. So the organism will not even notice that there is that type of mutation. Okay, so like we said, they have no impact on the organism's life. So it has no impact on the organism's life. In fact, the organism may not even notice this mutation. But what about beneficial mutations? Remember, beneficial mutations, we said that they are harmless. They do not cause any disorders. They do not cause any discomfort. They do not cause any inconvenience. They can either be neutral, where the organism does not even notice that it is there because they are not expressed or seen on the body of the organism, or they might give you features. So these mutations have a positive impact. They might provide features to the organism's body that have a positive impact on the organism's body and then on the organism's life. Okay? So, in that they increase the chances of survival. Wouldn't that be nice? They are in fact expressed or they show on the organism's body. Examples in humans include resistance to diseases such as AIDS and COVID-19. So there are people that might have the HIV virus living in their body. However, their body is resistant to that HIV virus. Therefore, they are HIV positive. Remember, being HIV positive does not mean that you are sick. HIV is the virus, the pathogen that causes AIDS. So those people that are HIV positive might not even develop the disease, which is AIDS. So the HIV virus is basically useless. It has no effect that leads to the disease AIDS. Same goes with COVID-19. Those people might have the COVID-19 virus in their body. However, that virus does nothing to them. Okay, this is a beneficial mutation because it increases the chance of survival. Another beneficial effect, example of a beneficial effect is increased fitness. There are those people that have a greater cardiovascular endurance or a greater muscle tone, or can be taller than the rest of the people. All of this is not harmful. All of this is beneficial, and it has a positive impact on the life of the organism. Okay, on part 2 on this lesson, we will be looking at DNA profiling. First question, what is DNA profiling? When we speak about DNA profiling, we are speaking about a technique or a method used to prepare a pattern of black bars on an X-ray film representing DNA fragments of a person using biotechnology. Now, biotechnology is a technique, or biotechnology is a science where fragments of an organism are used in order to make a product. Okay, so when we look at the definition of DNA profiling, what the scientists are doing is that they are using our DNA fragments, okay, to prepare a pattern of black bars on an X-ray film, and those patterns of black bars are used to represent DNA fragments of a person. So what is a DNA profile? Okay, DNA profiling is the technique or the method, okay, of preparing a pattern of black bars on an X-ray film representing fragments or pieces of a person's DNA using biotechnology. The technique or the method is DNA profiling, all right? But a DNA profile is a pattern of black bars on an X-ray film representing DNA fragments of a person. So the actual pattern of black bars is the DNA profile. The technique or the method used to prepare these pattern of black bars is called a DNA profile. So that is what a DNA profile is. Okay, so here we have an X-ray film, as you can see over there, that has a pattern of black bars or a pattern of black bands. So this is a DNA profile. However, the technique or method used to prepare these DNA profiles is called DNA profiling. Okay, so as a matter of interest, let us look at the following slide. It contains a very short clip or video to show how these are prepared.
First, technicians extract the DNA by dissolving the evidence. At that time, they needed a sample as large as a quarter to generate a DNA profile. Then, Jeffries has to pinpoint the DNA fragments that are unique to their owner. To do that, he uses a technique called RFLP, Restriction Fragment Length Polymorphism.
And what this does is that it cuts the DNA into sections of various sizes.
To get the DNA portrait he needs, Jeffries has to sort the fragments by size. Then the sample goes onto one end of a gel plate. Technicians apply electrical current, and electricity pulls the fragments through the gel.
The gel acts exactly like a sieve, and the small fragments are able to travel quickly through the gel, and the large fragments travel slowly. At the end of the process, we have an X-ray film, which effectively has a barcode on it.
It may look like random splotches, but that barcode is unique. In all the world, it belongs to only one person.
Okay guys, on the previous slide, we looked briefly at how a DNA profile is made. So on this slide, we will be continuing on our content of DNA profiling. We will be looking at the uses of DNA profiling, as well as some of the factors that affect the reliability, the trustworthiness. How can we trust, or how much should we trust DNA profiling? So firstly, let us look at some of the uses of DNA profiling. One is to identify relatives, okay? So we use DNA profiles, those patterns of black bars, to determine if somebody is your father, that is paternity, if somebody is your mother, that is maternity, if you are related in terms of parentage to that other person. That is determining if somebody is your siblings, okay? Your brother, your sister, and the likes. All right. Another use is to identify dead bodies. Usually, to identify a dead body, if you have received news that somebody has died, then the family members usually go to the mortuary, where they are going to identify the person by looking at their physical features. They will look at their face to identify if this is a deceased person that they know, okay? However, there are some instances where people die in a way that they become unidentifiable by looking at their bodies. For example, if a person is burnt beyond recognition, or if a person was involved in an accident, let's say for example, pardon my very graphic example, but if somebody has been run over by a train and their face is completely squashed, and that person, they found only the body parts of that person, and that person is unidentifiable. In those instances, then we may use DNA profiling in order to determine. We look at their DNA, the sequence of black bars. We compare that to a sample of a DNA profile that we already have of that person, and we can identify whether it is that person that has deceased or not. Okay, like I said, pardon my very graphic examples. We can use DNA profiling. It is also used as biological evidence in a criminal case. Okay? Please do not say it is used to identify a criminal. Okay, guys? Just because you prove that somebody was with let's say, for example, a dead person, before they had deceased, if you find their DNA on the body of that person, doesn't necessarily mean that they are the murderer, okay? That only proves that that person was with the deceased person or the victim of a murder before they had deceased. That does not necessarily mean that they are the murderer, okay, guys? So, to gather evidence in a criminal case, the court may use DNA profiles as biological evidence to build proof of a murder. Okay, guys? And then DNA profiling is used to diagnose inherited disorders. When you talk about inherited disorders, we are talking about disorders that one might inherit from their mother and their father. So, doctors may use DNA profiling to diagnose these genetic disorders, to say this person has this inherited disorder or the other inherited disorder. Another use for DNA profiling is to find cures for inherited disorders. Okay, so DNA profiling may be used to diagnose genetic disorders or inherited disorders, and they might be used to find cures for certain genetic disorders. Okay. All right, with the uses of DNA profiling, let's say, for example, we are using DNA profiles in a criminal case as biological evidence. Should we trust this DNA profiling? Is it a method of building evidence that we can absolutely trust, that is absolutely reliable? The simple answer is yes. Okay, the simple answer is yes. DNA profiling is very, very, very reliable, and it has been used as biological evidence in a criminal case, or it has been used to determine whether somebody is somebody else's father for a while now. It is very reliable. However, it is not 100% foolproof. It might be 99.9% reliable or trustworthy. However, there are certain things that compromise the reliability or the trustworthiness of DNA profiling. And if you get a question like that in the exam, here should be the answers. The answer. What are some of the arguments against DNA profiling? Or the question might be asked in a different way. What are some of the factors that reduce the reliability or trustworthiness of DNA profiling? So here are the answers. Okay. Because these DNA profiles are made by humans, okay, and they are also interpreted by humans, there is space for human error. So people working with DNA profiles may make mistakes. So when we talk about human error, we are talking about making mistakes either during the preparation of the DNA profiles or during the interpretation of the DNA profiles. So people working with DNA profiles may make mistakes during the process of preparing and interpreting the DNA profiles. Okay, so human error is our first factor that may reduce the reliability of DNA profiling. Secondly, it has to deal with corruption. Okay, people working with DNA profiles may accept bribes to falsify the results. This is not a nice thing to do. We know that. We live in South Africa where corruption is a major problem. Okay, so in a criminal case, for example, or a case that is dealing with paternity, if a person has money, then they may pay the technicians or they may pay whoever is working with the DNA profiles to falsify the results and render them false. But in actual fact, they were guilty. Okay. Thirdly, incorrect sampling. Since DNA is a long molecule, lab technicians may sample a piece of DNA at a different position from the one they are testing against. So like we said, DNA is about 5 meters, 4 meters, some sources say 7 meters long. So if you find DNA at a crime scene, for instance, and then the technicians may sample the first, let's say the first 3 or 4 centimeters, the first centimeters of the DNA. Okay, but the DNA of a criminal that is behind bars, that is suspected, sorry, not criminal, but the suspect that is being held in jail and they are suspected of being the murderer. On his DNA, they may sample the last 3 centimeters. But from the DNA that was found on the crime scene, they sampled the first 3 centimeters. So if there is incorrect sampling there, we may get inconsistent results, okay? And if we get inconsistent results, then that means that because of this incorrect sampling, then the validity of the results will be compromised. So we might get a different sequence of black bars on the DNA profiles because we sampled the first 3 centimeters and the last 3 centimeters on a different sampling. I hope this makes sense. Then the last one that we are going to talk about here, no, not the last one. The second last one is the expense. Okay, conducting tests through DNA profiling is expensive. Not everyone can afford it. This makes it accessible to some people. Okay, this is particularly applicable to civil cases. Civil cases such as when, for example, a mother is suing a particular gentleman for maintenance of a child. Okay, so in that case, the onus or the burden is on the individual person to do the paternity test using DNA profiling. Okay, they have to prove themselves that the other gentleman is the father. So they would have to pay for this procedure of DNA profiling. And these procedures cost anything from 4,000 Rand, 5,000 Rand, and above, depending on the lab. Okay, so the state does not pay, in civil cases, the state does not pay for the DNA profiling. The people involved in this dispute in court would have to pay this. So if you don't have the money to prove that somebody else is the father, unfortunately, you might lose the case. So it being expensive and only accessible to people with money makes it unreliable for a set population of people that cannot afford DNA profiling. And then lastly, inconsistent methods. What do we mean by this? Different labs use different methods of conducting the tests of DNA profiling. This therefore compromises the validity of the results as they are not standardized. Okay, so of course, if different methods are used by different laboratories to conduct these procedures of DNA profiling, because there are inconsistent ways of conduct or methods of conducting the tests, therefore it compromises the validity of those results. So sometimes because of this, people would actually want to do the DNA profiling tests again with a different lab and again with a third lab, okay, just to make sure that the reliability is not compromised. Okay, guys, so on this slide we discussed the uses of DNA profiling. They are prescribed for you to know. You must know them. And then we have also discussed some of the factors that affect the reliability or trustworthiness of DNA profiles. Okay, these lists are not exhaustive, meaning it is not all the uses and it is not all the factors that affect the reliability, but these are the main ones. Okay, now we are going to be doing some interpretive work on determining certain individuals using DNA profiling. So there are two types of scenarios that you might get in the exam, okay? You might be tested on, firstly, paternity. When we talk about paternity, we are talking about testing who is the biological father of a certain individual. And you will be tested again on biological evidence in determining who a criminal might be in a criminal case, in a murder or a rape case. All right. So in both scenarios, you are required to analyze the samples of DNA profiles, compare them, and conclude on which male is the biological father of the child in question. So that is scenario number one. Or you might be examined on to determine who a criminal might be in a criminal case. So this is scenario number two. Okay, so on the following slides, we will be doing interpretations on both scenarios. Okay, guys, let's start with our first scenario, testing for paternity. When you are testing for paternity, you will be given the following DNA profiles. The first DNA profile that you will be given is the DNA profile of the mother. Okay, remember, we are testing for paternity. We are testing who is the biological father of that particular child. However, you will be given a DNA profile of the mother as well. Why is this DNA profile of the mother given if we are testing who the father is? This sample acts as a control sample. This is the sample of the known parent. It is included in the investigation to make sure that it is the correct child that is being tested. And 50% of the black bands or black bars of the mother should match the black bars or the black bands of the child's DNA profile. So this is given to make sure that it is the correct child that is being tested. Okay, I will give you a scenario. For example, we have a mother that is alleging that her child was fathered by Tsepo, for example. Then the mother knows exactly that Tsepo is not the father of her child. Okay, however, the mother wants to trick Tsepo into supporting the child throughout its life. Okay, so the mother might take a DNA sample of Tsepo's biological children and submit that to the lab to be tested, okay, as to determine that Tsepo is the biological father. So if the mother takes the DNA sample of Tsepo, one of Tsepo's biological children, then the results are going to come, and then they will definitely say that Tsepo is the father. Okay, but if the same sample was analyzed and we checked for who the mother is, then it will be determined that she is not the mother. She is not the mother of Tsepo's children. Okay, so in order to close this gap, or in order to make sure that this does not happen, the DNA profile of the mother is included to make sure that the correct child, meaning the child to which is the child of the mother, is the one that is being tested on to determine who the father is. Okay. The second DNA profile that you will get, of course, is the DNA profile of the child. This is the DNA profile that is being analyzed against the samples of the identified males to determine which one of the males is the biological father. Then you will receive the DNA profiles of the multiple males. If the mother is unsure who the father is, male 1, male 2, male 3, you will receive DNA profiles of male 1, male 2, male 3. You are going to check the DNA profiles of each male against the DNA profile of the child, and the one that has 50% of the black bars that are matching to the black bars of the child's DNA profile, that is going to be the biological father. All right. So here we have a DNA profile. Our job is to study this these set of DNA profiles and determine who the father of the child is. Like I said, we have the DNA profile of the child, and then we have 3 DNA profiles of alleged fathers: male 1, male 2, and male 3. Okay, so from these 3 DNA profiles, our job is to analyze these 3 DNA profiles against the DNA profile of the child to determine which one of the 3 males is the biological father. Okay, guys? So where do we start? Okay, remember we said that the DNA profile of the mother is also included in this investigation, why, like we said, 50% of the child's DNA of the child's black bars must match 50% of the mother's black bars in order to make sure that it is this mother's child that is being tested and not tricking the system. Okay, so let us firstly start there. Let us determine which black bars of the child match that of the of the mother. Okay, so what you will do, you will take your ruler and check the pattern of black bars of the child's DNA profile versus the pattern of black bars of the mother's DNA profile. We want to rule them off because, like we said, 50% of the child's black bars must match the black bars of the mother, and 50% must match the black bars of the father. So we want to rule off the pattern of black bars that match with the mother. The ones that are left, we are going to study them against the ones of the males. Okay, so this black bar, when we check, the first black bar of the child does not match the black bar of the mother, okay, but the second one matches the black bar of the mother. Okay, so you are going to take your pen and then let's say a blue pen, and then we rule that off. There, it is a match. The third black bar also matches that of the mother. Okay, also matches that of the mother, right? When we check the third one, there's no match. The fourth one, there's no match. The fourth one, there's no match. The fifth one, there's no match. But the last one, there is a match. And then you rule it off as well. Okay? So we have 3 black bars that match with the mother. Okay? So the ones that are left, this one, that one, should match one of these one of these males. Okay, so let us check by using a different color. I'm going to use green to analyze. Let's check. This first black bar does not match male A. However, it matches male 2 and male 3. So there is a match there. There is a match there with that one black bar of the child. Okay, now the third one, the fourth one, the second one and the third one, those we are not going to look at because those are the black bars that match the mother. So we ruled them off. Okay, we go to this one over there, the second one. Okay, the third one, sorry. So when we look at the third one, this one over here, it matches with male 1 and it matches with male 3 over there. Okay, now we go to the following one. This one over there, it doesn't match with male 1, but it matches with male 2 and it matches with male 3. Okay, and then the last one over there, we have ruled off because it matches with the mother, meaning it is the segment of DNA that matches with the mother. So we are not going to analyze it with the father. Remember, 50% must match with the mother. So the 50% we've already ruled off is that bar, and that bar. So when we are comparing with the fathers, with the males, we do not include the ones that match with the mother. We include the ones that are left. Okay, so when we check, we are now supposed to analyze. And write down how many black bars match with each male. Right, we had one match with male 1. We had 2 matches with male 2. However, we had 3 matches with male 3. And remember, with the mother, we also had 3 matches. So let us count how many black bars of the child's DNA profile do we have. We have 1, 2, 3, 4, 5, and 6. 3 of them match with the mother, that is 50%, and 3 of them match with male 3, that's the other 50%. So the biological father is definitely male 3. This child might be related to male 2 and male 1, maybe the uncle, maybe the close cousin, but not the biological father. Okay, so the biological father in this instance is male 3. So that's how we analyze the DNA profiles when we are determining paternity. When we are determining paternity, 50% of the black bars or the black bands of the child's DNA profile must match that of the mother, and the other 50% of the pattern of black bars must match that of one of the alleged fathers. Now for question number 1, identify the technique shown above. The technique. Okay, guys, the technique or the method shown above. So the technique is a DNA profile. Okay? DNA profiling. The technique is DNA profiling. Okay? Which male is the biological father? This is easy. We've already determined that. The biological father is male 3. The following question is a bit obvious. Explain your answer to question 1.2. Okay? For 3 marks. Now, what you will not say is that the DNA of male 3 matches the DNA of the child 50%. Do not say that. We see that response a lot at the marking center, but it is incorrect, guys. Why is it incorrect? When we're looking at this given diagram, what we see here is not the DNA. You don't see the DNA when you're looking at this. What we in fact see, we see a pattern of black bars of the child, of the child's DNA profile, a pattern of black bars of the mother's DNA profile, a pattern of black bars of male 1, male 2, and male 3's DNA profiles. That's what we see. We do not see DNA. So you will not say that the DNA of male 3 matches with the DNA of the child, because what we see is a pattern of black bars on the DNA profiles, not the DNA. So please refrain from giving that response. Okay, how you are supposed to answer this is like this. 50% of what? Of the pattern of black bars or the pattern of black bands, you can say black bars or black bands, found on the child's DNA profile. So 50% of the pattern of black bands found on the child's DNA profile, so this DNA profile, matches the pattern of black bands on male 3's DNA profile. Okay, guys? So what we are seeing, like we said, 50%, it's supposed to be a mark there. There's supposed to be a mark there. 50% of the pattern of black bands found on the child's DNA profile matches the pattern of black bands on male 3's DNA profile. That's how you're supposed to answer this question, by referring to the pattern of black bands, not the DNA. Which is more than male 1 and male 2's matches? Okay, So we have 1, 2, 3, 4 marks. However, the question is out of 3 marks. Therefore, any 3 of these would suffice. Or how you would answer this question is, there are more black bands on male 3's DNA profile. Remember, we have 1, 2, 3 that are a match. Male 2 is only 2 and male 1 is only 1 match. So there are more black bands on male 3's DNA profile that match the pattern of black bands on the child's DNA profile than those found in male 1 and male 2. Okay, so personally, I prefer this answer over the option, this one. However, if you write the answer in this way or that way, you will get it correct. Okay, guys? So I've given you 2 ways in order to answer this question. You will always answer this question like this. If you are given a different set of DNA profiles, maybe the father on the question that you get is male 2. What you will replace is male 3 for male 2. But the answer will always be like this. So please do familiarize yourself in how we answer this question, because it will always be answered like this. It's one of those questions that you just need to know the skill of how you answer that. For the last question, the question says, state 2 other uses for this technique. 2 other uses for this technique. So the focus is here, other uses for this technique. Okay. Now, when you give a response to this question, you will not say to identify relatives. Okay. Why? Because in this instance, we were determining who among these 3 males is the biological father of this child. Okay, now identifying who a father is to a child is determining a relative, because a father is a direct relative of the child. So in this instance, we were determining a relative. And which relative were we determining? Who amongst these 3 males is the biological father to this child? Okay, so the question is saying state 2 other uses for this technique. If you write to determine relatives, it will be wrong. It will be wrong based on the fact that we were determining a relative to this child. We were determining who their biological father is. So you can give the following responses. To identify dead bodies, this would be correct, because remember, in the previous slides, we said that sometimes people die in such a tragic way where they cannot directly be identified facially. Maybe they are burnt beyond recognition. Maybe they were run over by a train, or maybe the dead body was discovered way after some time where the facial features had already decomposed. Okay, so in that instance, we may use DNA profiling to identify the dead body. Okay, the second one, used as biological evidence in a criminal case. So, in this instance, we are going to determine who a possible murderer is or who a possible rapist is by taking evidence that was found at the crime scene, making a DNA profile from it, and making DNA profiles from possible suspects, and we compare them against each other. The following one, to diagnose inherited disorders, and the last one, to find cures for inherited disorders. Okay, guys, so any 2 excuse me, sorry for that, any 2 of these 4 would get you the 2 marks. All right, but not the one of identifying relatives, because the question says 2 other uses for this technique. So on the second scenario, here we are going to be doing interpretation of DNA profiles, but the second scenario we will be testing for a possible, not a possible suspect. It's not supposed to be possible suspect there, excuse me, made an error. A possible criminal from a set of suspects. Okay. Now, which DNA profiles do we expect to find? So let's say we have a scenario where we have a dead body and the police have determined that it was a murder. Okay, guys? That it was a murder. The person was murdered. Okay. So what the police would do is that they would block off the area, the crime scene, and the forensic specialists would come and they would take samples of evidence in that crime scene. And then after that, they would take that to the lab where they extract DNA samples. And then, if there are maybe 4 different suspects that are in jail and they've been identified as suspects and they are on trial, what would happen is that each person's DNA sample from their bodies, those 4 persons, they would take DNA samples from those 4 persons and conduct DNA profiles. Okay, so the DNA profiles will be provided as evidence at court. So it will be suspect 1, suspect 2, suspect 3, and suspect 4's DNA profiles against the DNA profiles, of the DNA that was found on the crime scene. So they would match the DNA profiles of the evidence that was found at the crime scene versus the DNA profiles of the suspects that are jailed and undergoing trial. And then, in this instance, it's not supposed to be 50-50, right? The match must be 100% match of the black bars. Okay, but remember, like I said, it doesn't mean if the DNA profile of the evidence that was found in the crime scene matches 100% of suspect 1's DNA profile, for example. It doesn't mean that he is the murderer or she is the murderer. It doesn't necessarily mean that, because the person might have hugged the victim of the crime, or maybe they were together just before they died. That doesn't mean that they killed them. Okay, guys? So, you are going to be given DNA profiles. The first DNA profile that you'll be given is the DNA profile of the potential criminals. The DNA profiles of the potential criminals, meaning the DNA profiles of the suspects. Okay. This sample may be attained from a murder weapon, semen swab from the vagina of a rape victim, or skin cells under the nails of a dead body, etc. Okay, so this is the DNA profile of evidence found at the crime scene. Second DNA profile is a DNA profile of the victim themselves. Okay, now remember, here we are trying to determine who among the suspects is maybe the murderer, for example. But the DNA profile of the victim is also included maybe included in the investigation as well. Why? This sample acts as a control sample. It is included in the investigation to make sure that the DNA of the potential criminal is not that of the victim. Okay? So we're using it to isolate the DNA of the other people that might be the criminals. Okay? We are isolating it. It is used to isolate the DNA profiles of the other people. So if we know what the DNA if we know whose DNA profile is not of the criminals, we can focus on the other DNA samples, okay, and identify them as those that belong to the potential criminals. Then we are going to find the DNA profiles of the arrested suspects. These samples are those of the people who are suspected of having committed the crime. The suspect whose DNA, whose sequence of black bars match approximately 100% of the black bars of the DNA found at the crime scene is most likely the criminal. Like I said, it's not automatic that they are the criminal. Okay, guys? Okay, let's study the diagrams. Before we look at the questions. So we have a DNA profile there of the victim. Okay, remember we said that we include the victim's DNA profile. We include the victim's DNA profile to isolate the DNA of herself and the DNA of the potential criminal. Okay, and then we have the DNA that was found on the knife at the crime scene. So this is the murder weapon. Okay, so we have DNA maybe on the shaft or on the handle of the knife there. So it means that the DNA that is found there is most likely the DNA of the person who stabbed the criminal. And then they created this DNA profile according to the DNA that was found on the handle of the murder weapon. And then we have the DNA profile of these three gentlemen that were arrested and suspected of having murdered the victim. Okay, guys? Now here, the DNA profile of the suspect must match 100% to the DNA that is found on the DNA profile of the DNA that is found on the crime scene. Okay, guys? So here is not 50-50 because that DNA is the DNA of one of these gentlemen and it must match 100%. It's not like testing for paternity where only 50% of the black bars must match. Okay, let's determine who the murderer most likely is. So when we analyze the pattern of black bars, these pattern of black bars, when we check them, let's check them one by one. This black bar does not match this suspect. It matches that suspect and not match this suspect. So automatically, if we don't find a match with the first bar, we rule off suspect 1 and we rule off suspect 3. They are most definitely not the murderers. Okay? Because one of their black bars does not match. Okay? But do not just rule them off based on one bar, guys. Rule them off based on all of the bars. Okay, now when we look at this first bar and we look at that bar, it matches. The second one is a match. The third one is a match to suspect 2. The third one is also a match to suspect 2. The fourth one also matches that of suspect 2, and then the last one also matches that of suspect 2. So we submit that suspect 2 is most likely the murderer or the criminal. Let's look at our first question. Which suspect is most likely the murderer? The answer is suspect 2. You can almost determine what the next question is going to be. Of course, the next question is going to be provide a reason for your answer to question 1.1. And this is how you answer the question. Remember, the answer will not be the DNA of suspect 2 matches 100% to the DNA of the evidence that is found at the crime scene. Remember, you do not see the DNA here. However, what we do see is the sequence of black bars or the sequence of black bands of the evidence that is found at the crime scene and of the DNA profile of suspect 2. So the answer is the pattern of black bands of suspect 2's DNA profile matches 100% with the pattern of black bands of the evidence of the DNA profile of the evidence found at the crime scene. This is not supposed to be 1 mark, guys. This is not supposed to be 1 mark. It's supposed to be 3 marks. Pardon me for that error. So the pattern of black bands of suspect 2's DNA profile, that's where the first tick is supposed to go, matches 100%. That's where the second tick is supposed to go, with the pattern of black bands of the DNA profile of evidence found at the crime scene. Hence, it is going to be 3 marks. The following question: why is the victim's DNA profile included in the investigation? Remember, we are determining who is most likely the murderer. Okay, so it makes sense that suspect 1, 2, 3's DNA profiles are included and the DNA profile of evidence found at the crime scene. However, why is the victim's DNA profile included? The answer is this sample acts as a control sample. It is included in the investigation to make sure that the DNA of the potential criminal is not that of the victim, just like we discussed in the previous slide. Or you can say this sample acts as a control sample. It is included in the investigation to distinguish or differentiate between the victim's DNA and the suspect's DNA. Okay, guys? The following question: provide and explain 2 arguments that the lawyer representing the suspect you identified in question 1.1 can make against this evidence. Okay, remember, in a criminal case, suspect 2 would be represented by a lawyer, an attorney, or an advocate. So this is a cognitive level 4 question, right? It requires you to do some thinking and application to the knowledge that you already know. Okay, so what may this lawyer argue against the evidence that his client, suspect 2, is the potential criminal? Okay, now, how do we relate? Or what do we relate? What do we reference? Which content do we reference in order to answer this question? It is the content on the factors that affect the reliability of DNA profiling. Okay, and what did we say are the factors that affect the reliability or that reduce the reliability of DNA profiling? The first one, the lawyer might argue, people working with DNA profiles may make mistakes during the process of preparing and interpreting the DNA profiles. 1. It's supposed to be 2 marks. It's supposed to be 2 marks. So 2 ticks there. Or they may argue, people working with DNA profiles may accept bribes to falsify the results. This is another 2 marks, 2 ticks. Or you may say, since DNA is a long molecule, the lab technicians may sample a piece of DNA that is at a different position from the one they are testing against. This is an additional 2 marks. Or you can say, conducting tests through DNA profiling is expensive and not everyone can afford it. Okay, now in a criminal case, this one does not apply. Okay, this one only applies to civil cases, for example, when you are testing for paternity. So this one does not apply. So any two arguments of these three is going to suffice. And then let's look at the last question. Suppose that the government of South Africa is considering to collect DNA samples of every citizen in the country to compile a database and store everyone's DNA profile to have them as biological evidence for any court case. Provide one argument for and one argument against this proposition. Okay, so if the government of South Africa, for instance, wanted to, every single individual, every single citizen in South Africa, they take their DNA samples and then they prepare DNA profiles for everyone in the country, and then they store their DNA profiles in a database. Okay, what one argument can you make for this, and what one argument can you make against this? Okay, so this is not a content-based question. It is a cognitive level 4 question that requires you to think and make sense of the scenario that you are given. So what can you argue? Okay, the arguments may be: 1. Arguments for this proposition. The DNA profiles will be readily available for cases on trial, meaning at court. The trials will run faster. So just like fingerprints, right? The Department of Home Affairs has all our fingerprints. So if the police are running an investigation and they find fingerprints at a crime scene, they will take those fingerprints and compare them to the fingerprints that they already have from the Department of Home Affairs. Okay, so just like that, if the government already has DNA profiles, and then there's a murder trial, they would already have these DNA profiles in their database, and if they get a suspect, then they conduct a DNA profile on those suspects and they pin them against what they already have in their database. The trials will run faster instead of when they have to do new DNA profiles from scratch. Another argument: this project requires a lot of people. It will provide more job opportunities. It will provide more job opportunities, especially in South Africa, where we know we have a problem with poverty and a problem with unemployment. Okay, so because this is going to be a major project, we are going to require more people. Lab technicians that are unemployed will be hired. Field workers that are going to collect DNA samples from homes of different people will be employed. Analysts of these DNA profiles will also be employed. Drivers that are going to drive these samples in and around the labs and the likes will also be employed. So since this is a major project, it requires a lot of people and it will provide more job opportunities for those that are unemployed. What about arguments against? Remember, the question requires you to provide one argument for and one argument against. Okay, guys? Therefore, one of these two will suffice. Now, let's look at argument against. You may answer to say South Africa does not have the budget or enough resources to run this project, as it is expensive to start and expensive to maintain. Okay? Money would have to be taken from other people and departments to supply for this project. Okay, so since South Africa does not have the money to start and maintain this project, it means that there is going to be restructuring of the budget, and then money would be taken maybe from the Department of Education or from the Department of Health or from any other department, and then will be invested into this project. And that would seem unfair in certain ways. Okay, or you can argue to say this will be a violation of human rights. DNA profiling may reveal private information about individuals, such as inherited disorders. Okay, so DNA profiling does not only reveal what the Department of Justice might want it to reveal, it might also reveal things about the individual that are not, that the individual does not intend on revealing. And remember, since this will be a database, it will be on public record, therefore the information will be out there for everyone to see. So this is an argument that you might make against this proposition that may be made by the government. Okay, guys. All right, guys, this is the conclusion of our lesson 7 of the lesson series on the DNA Code of Life. Please, if you've been enjoying my content or if you've benefited in any way on either one of the videos, please like, comment, subscribe, and share the link to this video so that it may increase the reachability to different people and also grow my channel. It is a small click for you, but for me, it means the world. As usual, thank you so much for watching. The next lesson is on meiosis. It's a complete new lesson on a complete new topic of meiosis. It is going to be on topic 2 of your academic year, and I will be including it in a completely new series. So be sure to check that series out. As usual, I will be providing detailed lessons on that particular topic. Thank you so very much for watching, and thank you so very much for your continued support. See you next time. Bye-bye.